Hello, my name is Joshua Gabi. I welcome you to Meiosis. Lesson objectives. Today we are going to look at sexual life cycle, gametes, stages of meiosis. We shall look at crossing over, random assortment, non-disjunction, and karyotyping. You are welcome. Please don't forget to subscribe. Sexual life cycle. Sexually reproducing organisms inherit genetic information from both parents. When we are looking at sexually reproducing organisms, these ones, they inherit genetic information from both parents. This means that some of the information comes from the maternal and the other information comes from the paternal side. This results in offsprings with 2 copies of every chromosome. So we shall have 2 copies of every chromosome, and that is what we call a diploid. We shall have 2 copies of every chromosome, 1 chromosome from each parent. In order to maintain static chromosome number across generations, a diploid organism must have the chromosome number of their sex cells, i.e. haploid. So this means that if we are to maintain a static or the same chromosome number from generation to generation, an organism or a parent, maternal or paternal organisms, they have first of all to have half the chromosome number of their sex cell to make them haploid. And now that is what we shall call the formation of gametes. The resulting fusion of these haploid sex cell, which we call the gametes, creates a diploid cell that can multiply and differentiate to form the entirely new organism. So we are having haploid cells, which are the gametes, coming from the maternal and another one coming from the paternal. When they fuse or combine together, they create a diploid cell. Now it is a full cell. The other ones were haploid, half. They were halved. Half the chromosome number, that is the sex cells, were half the chromosome number, which we call the gametes. For example, the sperm is half the chromosome number, and the ovum is half the chromosome number. So when they fuse, then they create a diploid cell, which we call a zygote. It is now having full number of cells. Now that one is what we call the diploid cell. And now that one can now multiply and it can differentiate and it forms a new organism. The offsprings will be genetically amalgam. It will be a genetic amalgam of both parents, but inherently distinct. The halving of the chromosome number allows a sexual life cycle with the fusion of gametes. So when we have the chromosome number, this one allows the sexual life cycle with the fusion of gametes. Now, if we are fusing the gametes, that means that the cells of the gametes, the chromosome number should be a half, because you have half the number in the sperm gamete and half the number of chromosome in the ovum gamete or the egg gamete. So when they fuse together, now they form the full number of chromosomes, which makes it to become a complete, or what we call a diploid cell. Let's now look at the gamete. Gametes, these are haploid sexy cells produced by sexually reproducing organisms. What we call the gametes, we are saying they are haploid cells, cells which have half the number of the total chromosome of a somatic cell. So we have the female gamete is what we call the ovum. This is our ovum. And the male gamete is the one that we call the sperm. All of these cells, they are haploid. The human egg or the ovum has half the number of chromosome, and the other half of the number of the chromosome is in the human sperm, or what we call the spermatozoa. So they are haploid. The gametes are haploid. The female produces eggs, while the male produces sperms. Haploid cells are created via a process we call meiosis, and then they differentiate via gametogenesis. So during meiosis, this is a cell division which takes place and results in the formation of 4 haploid cells, which are half the number of the total chromosome number, and that is meiosis. Now, after forming these cells, then they are going to differentiate into what we call the eggs via gametogenesis. Meiosis. Meiosis is a process of making haploid sexy cells, or what we call the gametes. Meiosis, this is a process whereby we are going to form haploid sexy cells, which we call the gametes. It is preceded by interphase and involves 2 division. It starts with interphase, and in interphase, that is the phase where the cell is preparing itself for cell division, creating organelles, creating enough energy, storing enough nutrients to make sure that the cell undergoes the division. And it undergoes the first division, which we call first meiotic division, and then it undergoes the second division, which we call the second meiotic division. So if we start with our cell, like this cell we have up here, that is our first cell here. And if it is undergoing meiosis, there is that part which we are calling interphase. During interphase here, it is now the cell mobilizing itself. It mobilizes itself, chromosomes, they condense, there is a preparation of nutrients, there is preparation of energy, and then it undergoes the first meiotic division. Under the first meiotic division, we get these two cells. That is the first meiotic division. Then after the first meiotic division, then we go to the second meiotic division. Under the second meiotic division is when we get now our gamete cells, which are haploid, 4 of them, and that is what we call meiosis. The first division separates the homologous chromosome. That is during the first meiotic division. And we have seen that the first meiotic division takes place around here. And what we have inside are what we are referring to as homologous chromosome. Now, when the first meiotic division takes place, it is going to divide those the homologous chromosomes. They are going to be separated, and each one will go in its own cell. So that one is what we refer to as the first meiotic division, or the first division. It separates the homologous chromosome, and each chromosome goes into its own cell. It is a reduction division. It is a reduction division, meaning that now the cell is becoming haploid. It was diploid, but now the cells are becoming haploid. If you look at this first cell here, you will see that the cell is diploid. It has 2 pairs, or it has a pair of chromosomes. Now, when they undergo division and they go each one in its own cell, now that one means that we have now divided them. So which means that the number has reduced. From being 2 chromosomes in one cell, now we have 1 chromosome in each cell. That is what we are referring to as that it is a reduction division. It is a reduction division. Now it has moved from being 2 chromosomes in a cell to 1 chromosome in a cell, from diploid to haploid. Meiosis, we are still in meiosis. Now we have the second division. In the second division, this one separates the sister chromatid. Now let's go to the second division. Now we have our cells that we have got from the first division, these 2 cells. Now here we have a chromosome, and this chromosome that we have, it has 2 sister chromatid. When you look at it, it has 2 chromosomes. There are 2 chromosomes attached together, and we refer to them as sister chromatids. So now in the second division, these sister chromatids are going to separate themselves, and each chromatid will go in its own cell. So the sister chromatids are separated. That is during the second division. The sister chromatids will separate themselves, and each chromatid goes in its own cell. That is during the second division. And it is a mitotic division. It is a mitotic division of a haploid cell. Already here, these cells are already haploid. These cells are already haploid. So when we divide them to take them down to form our cells down, here we are just only mitotically dividing the haploid cell. It is a mitotic division. The second division is a mitotic division because the cells are already haploid. Each cell has half the number of chromosomes. But here we are just carrying out mitotic division. The end result is 4 genetically distinct haploid cells. We end up with 4 distinct haploid cells. So our cells that we have here, there are 4 of them, but distinct, different from each other. 4 distinct haploid cells. They are the ones that we end up with in the meiosis. Let's look at the first stage, interphase. Now, the DNA is replicated during the S phase of interphase. Now, during interphase, what is happening here? We have a single chromatid chromosome. We have a single chromatid chromosome. That is before replication. It is just a single chromatid chromosome. Now, when it makes a pair, a copy of itself, now that is what we are referring to as replication. It replicates, it makes a copy of itself. That is during the S phase. That is what we are calling replication. Now, from being a single chromatid chromosome, now it becomes a sister chromatid chromosome. It becomes a sister chromatid chromosome. That is post-replication. The DNA is replicated. That is during the S phase of interphase. The replicated chromosome will consist of genetically identical sister chromatids. Now, this chromosome that we have formed here, they are identical. It is a copy of the original. The original one is this first chromatid that we had. This first chromosome is the original. Now it has made a copy of itself, which we are referring to as the sister chromatid. But those 2 sister chromatids, they are identical. They are a copy of each other. That's what we are saying. Replicated chromosome will consist of genetically identical sister chromatids. Let's look at the stages of meiosis. Now, the stages of meiosis, we have prophase 1. That is during meiosis 1. We have prophase 1, then we have metaphase 1, we have anaphase 1, and then we have telophase 1. You can see that in prophase 1, the homologous chromosomes are aligning themselves up. We have homologous chromosome attaching themselves to the centromere. I mean to the centrioles, okay? String. And then here, the homologous chromosomes, now they align themselves at the equator. That is during metaphase. Now, in anaphase, the homologous chromosomes now are being separated. The homologous chromosomes are being separated and they are moving to opposite pole. And here in telophase, we end up with the chromosomes have reached the different poles, and then also cytokinesis takes place, whereby the cell now it is dividing itself into 2. That is the telophase. Now that is during meiosis 1. That is during meiosis 1. Now I want us to see here in meiosis 1, in metaphase 1, and also prophase 1, you can see that there is crossing over taking place at these homologous chromosomes. There is that crossing over. If you come to metaphase, you will see that these two homologous chromosomes, they are forming what we are calling crossing over. When you come to anaphase here, you will see that part of the other chromosome is taken by this chromosome, and the other part of the other chromosome has been taken by this chromosome. So that is what we call crossing over. And that one takes place in meiosis 1. Now, after meiosis 1, we go to meiosis 2. In meiosis 2, we start with prophase 2. Now, in prophase 2 now, we have distinct condensed chromosomes. We have our condensed chromosomes. They are now there. Even in the other cell, we have condensed chromosomes. Now, they undergo now to a second stage, which we call metaphase 2. In metaphase 2, the chromosomes, each one aligns itself on its own spindle. You can see that each chromosome is aligning itself on its own spindle, and they are at the equator. Now, after that one, when we go to anaphase 2, you will see that each chromosome, now it is dividing itself. The sister chromatids are dividing themselves. One chromatid is going another side, and another chromatid is going another side. If you look at them, the sister chromatids have divided themselves, and they are moving to different poles. Now, we end up by the sister chromatid reaching the different pole, and that is during telophase. In telophase 2, the sister chromatids have reached the different pole, and then now we shall start seeing cytokinesis taking place, the cell dividing itself in the center. The cells will start dividing themselves in the center, what we call cytokinesis. And we shall end up having 4 daughter cells. The stages of meiosis. Let's look at the first division. We move to the first division. Prophase. In prophase 1, that is the first meiosis, meiosis 1. What is happening? Now here in meiosis 1, the homologous pair form a bivalent. They form a bivalent. Now in prophase, when we are coming from interphase, whereby the chromosomes are very small, they are visible like thread-like structure. Now when they come to prophase 1, in prophase 1, now we can see that they are condensed. They can be seen as chromosomes, as homologous chromosomes, aligning themselves together. And when they form here a bivalent, there is crossing over occurring at a chiasmata. Now, if you look here, you can see that crossing over is taking place here. At that point where they cross over each other, that is what we are calling the chiasmata. The homologous chromosomes, they form bivalent, and crossing over occurs at that point called the chiasmata. Now, the chromosomes condense. If you look, when they were in interphase here, they were thread-like structure, very small and thread. But when you come now in prophase here, now they have condensed. They have become shorter and thicker. You can see that they are short and thicker. That is what we are saying, that the chromosomes condense. The nuclear membrane breaks away. You can start seeing that this nuclear membrane that you are seeing here, the nuclear membrane outside, now here it starts disappearing. The nuclear membrane, if you are looking, the nuclear membrane is breaking away and disappearing. So that's what we are saying. During prophase 1, the nuclear membrane disappears or breaks down. Now we go to metaphase 1. Metaphase 1, we are moving now from prophase 1. This is our prophase 1. Now in metaphase 1, now we shall see the homologous chromosome aligning themselves on the spindle, and they align themselves at the equator, that is metaphase 1. You can see this homologous chromosome here, they have aligned themselves on the same spindle. Even these ones, the homologous chromosomes, they align themselves on the same spindle, and they align themselves at the equator of the cell, in the middle of the cell is what we call the equator of the cell. So here they say microtubule spindles fiber connect from the centrosomes to the centromere. We are saying microtubule spindles fiber connect from the centrosome to the centromere. Where are the centrosome? Now these are the centrosome that you are seeing on the side. Those are the centrosome. And the microtubule 5 spindles are these lines that you are seeing that will go and attach themselves on the spindle. Let's look at the phase of mitosis 1 here. Then we can see it very clearly. Now we can see that our centrosomes are these ones. Those are our centrosomes. We have 2 centrosomes. And from the centrosomes, that is where the microtubules fibers come from. They come from this point, as you're seeing, those microtubules, and they come at the center of the sister chromatid. At the center of the sister chromatid, that is what we call the centromere. That center of the sister chromatid is what we call the centromere. So these fibers, which we are calling the microtubule spindles, will connect from the centrosomes and connect to the centromere of the chromosome. So they come and connect to the centromere of the chromosome and they pass through. Also from the other side, another microtubule spindle will come and connect to the centromere. The same applies to the other ones. The spindles will come and connect at the centromere of the chromosome. Spindles from the centrosome, they come and connect to the centromere of the chromosome. Then the spindle fibers now causing the bivalent to align at the center. As we are seeing here, we are seeing that now this is a bivalent, or what we are calling a homologous chromosome. They align themselves at the center of the cell. That's what we are saying, that the spindle fibers will contract, and when they contract, they start pulling the homologous chromosome and they make them to align at the center. They will cause them to align the center. The bivalent will align themselves at the center because of the contraction of the spindle fibers. That is in metaphase 1. Then from metaphase 1, we go to anaphase 1. In anaphase 1, the spindle fiber contraction is going to cause the bivalents to separate. So we are now from metaphase here, where they have aligned themselves on the spindle fibers, and they have aligned themselves at the equator because of the contraction of the spindle fiber. Now, as the spindle fiber contraction continues, the bivalent is this homologous chromosome here. They are going now to start separating. Now, if you look here, when we come now to anaphase 1, you will see that the bivalent have separated themselves. This one is going in a different direction, and this one is also going in a different direction. This one is going the other direction, and this one is going in this direction. So the bivalent chromosome, or the homologous chromosomes, have been separated and they are moving to different poles. So here we see that the homologous chromosomes, now they will move to opposite pole of the cell. What we are seeing here, this one is moving in that direction and this one is moving in this direction. Here this one goes the other direction and this one goes the other direction. So that's what we are saying, that the homologous chromosome are going to move in opposite poles. That is during anaphase 1. That is during anaphase 1. Now we go to telophase 1. In telophase 1, now the chromosomes have reached the different poles. Our homologous chromosomes have gone separate pole and now they have reached to different pole. Here they are at different pole and here they are at a different pole. Now what is going to happen is that now the cell is going to divide itself into two, what we call cytokinesis. It divides into two and we shall end up forming two cells. We shall end up forming two daughter cells, and that is what we call telophase 1. During telophase 1, the chromosomes now will start to decondense. They will start to decondense. Those ones that were condensed, which were thick, they will start now becoming very thin. And then the nuclear membrane is going to reform. You will see that the nuclear membrane will start forming around the chromosome. The nuclear membrane will start forming around the chromosome. And then, lastly, cytokinesis occurs concurrently at the same time. Cytokinesis is going to take place. Cytokinesis is that one whereby the cell now is dividing itself into two. It also occurs at the same time, and at the end we shall have two daughter cells that are completely separate from each other after cytokinesis has taken place. Now the daughter cells are now haploid. The daughter cells are now haploid. We have divided them, that is telophase 1. Now the daughter cells are haploid. Each one is having only one pair of a chromosome, one chromosome of the pair, one chromosome of the homologous chromosome. Stages of meiosis. Now we are still looking at stages of meiosis. We have finished the first meiotic division. Now we are going to the second meiotic division. Now in the second meiotic division, we are repeating the same cycles that we have gone in through the first meiotic division. But the issue here now, the first meiotic division, we were separating the homologous chromosomes. Now in the second meiotic division, we are just going to separate the sister chromatid. In the first meiotic division, it was division of the chromosome, halving the chromosome number. But now in the second meiotic division, we are just going to separate the sister chromatid. So in the second meiotic division, it is just mitotic division which is taking place. Now here in the first meiotic division, we ended up with forming two sister chromatids. We had two sister cells, two daughter cells. Okay, now these two daughter cells, now they are going to undergo prophase 2. In prophase 2, now we are going again to recondense the chromosome. The chromosomes are going to recondense. The other side, they are decondensed. They had become smaller and thinner. Now they are going to become thick and short and visible. We shall be able to see them. Then secondly, the nuclear membrane. The nuclear membrane is going still to break down. You will see that your distinctive nuclear membrane that you are seeing the other time, now it is starting to disappear. The nuclear membrane is disappearing and allowing the chromosomes to be free within the cytoplasm. And then the centrosomes, now the centrosomes are going to move to the different poles. Here, if you look, when we finished our first meiotic division, the centrosome were at the same side. The centrosome were at the same side. But now, in prophase 1, centrosome are going to move at different poles. You will see them moving at different poles. They will come to different poles. That is in prophase 2. Then from prophase 2, we go to metaphase 2. What is going to happen in metaphase 2? Now, our microtubule spindle fibers, these ones are going to connect the centromeres and contract. So when you look here, you will see that during metaphase 2, the chromosomes, the sister chromatids, they are going to be attached on the spindle. If you look at the spindle tubules, these spindle fibers, they are moving from the centromere and coming to they are moving from the centrosomes, they are coming to the centromere of the sister chromatids. They pass through the centromere of the sister chromatid, coming from the centrosome. From the centrosome, they come and pass through the centromere of the sister chromatid or of our chromosome. So that is what we are referring. That is what is taking place during metaphase 2. We are having the spindles moving from the centrosome and attaching themselves to the centromere of the sister chromatids or our chromosome. That's what's happening first. And then they start contracting. After that one, they start contracting. Now, when they start contracting, the chromosomes are going to align themselves at the center. So you will see the chromosomes aligning themselves at the center of the cell. That's where the chromosomes are going to align themselves at the center of the cell. What we are calling aligning themselves at the center. And that is the end of metaphase 2. Now from metaphase 2, we go to anaphase 2. We finished with metaphase 2 when the sister chromatids had aligned themselves in the center, the equator, or at the center of the cell. Okay, so now what is going to happen is that the spindle fiber contraction is going to cause the sister chromatids to separate, and they will move to opposite poles of the cell. Here we ended in metaphase. Here we saw that the sister chromatid had aligned themselves on the spindles in the equator or at the center of the equator, the center of the cell. Now what is going to happen in anaphase? These sister chromatids now they are going to separate and they start moving at different poles. That one will separate and move to different pole. The other one will separate and move to different pole. This one will separate and move to the different pole, and this one will separate and move to the different pole. So that's what we are saying, that the spindle fiber contraction, as the spindle fibers start contracting and they are contracting, moving away towards the centrosome, they are going to pull the sister chromatids. And when they pull the sister chromatids are going to separate and they will start moving to opposite pole. That is in anaphase 2. Then from anaphase 2 to telophase 2, the chromosome now will have reached the end. The chromosomes will have reached the end. We are from here. Now we are having our chromosome reaching the end, and now they will start decondensing. Our chromosomes have reached the end and they are starting to decondensing. That is, now they are beginning to become smaller and longer. Then after that one, the nuclear membrane is going to reform. We shall have a nuclear membrane reforming. You can see that the nuclear membrane has started reforming around the chromosomes, and they are now enclosed within the nucleus. They are no longer free in the cytoplasm. They are enclosed in the nucleus. That's what we are saying, that the nuclear membrane is going to reform. And then cytokinesis. Cytokinesis is also going to occur at the same time. Now, cytokinesis, it is whereby we are having now our cell dividing itself in the center. The cell is going to divide itself in the center. And then after dividing itself in the center, now we are going to have 4 haploid cells. We have now 4 haploid cells, distinct cells. 4 different cells now have been formed. 4 daughter cells now have been formed after cytokinesis. The summary of meiotic division. Meiotic division, meiosis is the division of a diploid cell to produce 4 genetically distinct haploid cells. So we have 4 genetically distinct haploid cells formed. And we started with a diploid cell, but we have ended up with a haploid cell. So it occurs only in sexy cells. It occurs only in sexy cells. Mitotic division occurs in sexy cell. Those are the ones that we form the gametes. And it involves 2 divisions. We have seen there is division 1, meiosis 1, and division 2, meiosis 2. And this produces 4 daughter cells. We have seen that we have ended up producing 4 daughter cells. And the daughter cells are genetically distinct. The daughter cells are genetically different from each other. So let's look at interphase, that is interphase. Now prophase 1, metaphase aligning themselves at the equator. Then we go to anaphase, moving to different pole, homologous chromosome. Telophase, they have reached at the pole. Cytokinesis takes place. That is meiosis 1. When we go to prophase 2, meiosis 2, metaphase 2 align themselves at the center. Anaphase 2, the sister chromatid move at different poles. And telophase 2, we end up forming 4 sister daughter cells. Meiosis versus mitosis. We want to look at meiosis versus mitosis. Now, here you are going to see that we have divisions. In meiosis and mitosis, we want to compare the 2. Division. In meiosis, we have 2 divisions. And in mitosis, we have 1 division. When it comes to inheritance, in meiosis, alleles, they segregate. That is in meiosis. And in mitosis, there is no segregation. Synapses, that is forming crossing over, it occurs in meiosis. In mitosis, there is no synapses. Crossing over is present in meiosis, and it is absent in mitosis. The outcomes, we have 4 cells formed, and in mitosis, we have 2 cells formed. The haploid, in meiosis, we move from diploid to haploid. In mitosis, it is diploid to diploid. The use, in meiosis, it is used to make sex cells. In mitosis, it makes the body cells. Genetics, here the cells in meiosis are identical cells, and in mitosis, genetic variation. Please don't forget to subscribe. Now let us look at the purpose of meiosis. What is the purpose of meiosis? The primary purpose of meiosis is to make haploid cell, and those are for sexual reproduction. That is the primary purpose of meiosis, is to make haploid cells, which we call gametes, and they are for sexual reproduction. These cells are genetically distinct, which promotes variation in offspring. The cells that we form, they are genetically different from each other. And because of being genetically different or genetically distinct, that one promotes or brings about variation in the offspring, or it brings about a difference in the offsprings that we are going to produce. There are three mechanisms by which meiosis promotes genetic variation. There are three mechanisms by which meiosis is going to bring about variation. Children are going to be different, though they are the same gametes coming from the same cell, but they are distinct, they are different from each other, and they're going to bring about variation. How does meiosis brings about that one? One, there is what we are calling crossing over, and this is between non-sister chromatids, and it takes place in prophase 1. Now here we are having the separation of homologous chromosome. Remember, when we talked about homologous chromosome, we say these are chromosomes that came from different parents. One came from the maternal and another one came from the paternal, and they are moving together. Therefore, we call them homologous chromosome. Now, when they are in prophase 1, they are going to be separated. They are aligning themselves on the same spindle. But now when the spindle fibers start contracting, they will start pulling these homologous chromosomes to different pole. And in the process of going to different pole, they might have caused a synapse somewhere. There might have been an attachment whereby one chromosome was touching another chromosome. And when they are moving, they break. And at this crossing over point, at the chiasmata, they break and take the part of another chromosome. That is what we are calling crossing over. Now, some of the information from this side is going to go with the other chromosome, and the other information from the other side will go with the other chromosome. So there has been a crossing over, and that one is going to bring about the genetic variation. Then the second one is what we are calling random assortment. Random assortment of homologous chromosome, and that one takes place in metaphase 1. Random assortment of homologous chromosome. What do we mean by random assortment? Now, in metaphase 1, we have seen that the homologous chromosomes are aligning themselves on the spindle in the equator of the cell at the center. Now, when they are aligning themselves, the homologous chromosome, this is a pair of chromosomes. One chromosome came from the father and another chromosome came from the mother. Now, when all the homologous chromosomes align themselves on the spindle in the equator, the way they arrange themselves, they don't arrange themselves that all those ones that came from the maternal side should arrange themselves on one side. All those ones that came from the paternal side, they arrange themselves on the same side, so that when there is going to be separation, they go in the same cell. No, it is going to be random assortment. Some will align when the maternal is on the left and the paternal on the right. Then the other pair can be maternal on the right and paternal on the left. So that is what we are calling random assortment. They align themselves differently. So that one, when they are being separated and going to the different pole, you will have some of the maternal cells going to the left and some of the paternal cells going to the left and others going to the right. So those ones are going to bring about genetic variation. Then the fusion of gametes. When it comes to the fusion of gametes, this one is also random. Fusion of gametes. Gametes can fuse with any gamete. There is no specific order that this gamete should fuse with this gamete. No, it is random. So also during the fusion of gametes, it is going to bring about genetic variation. So those are the three mechanisms by which meiosis promotes genetic variation. There is crossing over, transferring of material from non-sister chromatids. Then there is random assortment, arranging themselves in the way they want. And then the fusion of gamete. Fusion of gamete is going to bring information from the paternal and information from the maternal, which are different, and they are going to now to produce offsprings which are going to be different. So that is how meiosis brings about or promotes genetic variation. Now, crossing over and random orientation promotes genetic variation. Crossing over and random orientation brings about genetic variation, which we have called random assortment. Genetic variation improves the survival prospect. Genetic variation improves the survival prospect of the species, and it increases biodiversity. How does it bring about survival prospect? That because of this genetic variation, some of the characters may be adopted by the environment and others may be rejected by the environment. And because of this variation in the genetics, it can cause some of them to survive and then others will maybe be eliminated. So because of having different variation, it will help the species to survive because it has a variety. And this is what brings about what we call biodiversity. Biodiversity is having very many variations within the species. Sources of variation. We are starting with crossing over. Sources of variation, crossing over. Now, crossing over occurs via a process of synapsis, and this occurs during prophase 1. So we talked about prophase 1. In prophase 1, we have homologous chromosomes aligning themselves together, moving together. And you will find that this one might have come from the paternal and this one might have come from the maternal, but they are homologous chromosomes. They are moving together. And in that process of moving together, they may form what we call a synapse. A synapse is that one where they cross each other. The chromatid of non-sister chromatids, chromatids of different homologous chromosomes, they come and cross each other. And when they cross each other, then they form what we are calling the crossing over. And now when it comes at the end, you will see that now they have separated. And now this one has taken part of the other one, and then the other one has taken another part of this one. So that is what we are calling crossing over. And that one is now going to bring about what we are calling variation. There is going to be variation, and that is what we are calling crossing over. Secondly, homologous chromosomes connect via the chiasmata. Homologous chromosomes connect via chiasmata to form a bivalent tetrads. They connected after a charge matter. Now, our charge matter here, where we are causing the crossing over, that is our charge matter. They form a bivalent tetrad. They form a bivalent tetrad. 4 connected together. They are now connected together, bivalent tetrad. Genetic exchange at this charge matter, the genetic exchange at this charge matter results in a recombinant chromosome. So we are having this charge matter. They are going to exchange. And when they exchange, now they have exchanged at the charge matter. Now we have formed what we are calling recombinant chromosome. Now these ones are our recombinant chromosome because they have exchanged genetic material across the charge matter. Now these ones we call them recombinant chromosomes. So the early stages involves the pairing of homologous chromosomes. That is the pairing of homologous chromosomes. They are pairing. So here you have your homologous chromosomes pairing up together, and then crossing over is followed. Crossing over now takes place here. That is followed by condensation. Then still we are looking at the sources of variation. Now let's look at random assortment. Now this one occurs during metaphase 1, where the homologous chromosomes line up in a random orientation. They are lining themselves up in a random orientation. The homologous chromosome during metaphase 1, they align up themselves in a random orientation. Now, if you look up this one here, you have, if we have our parent cell here, you can see that we have the chromosomes. Here, our chromosome. This one is homologous to that one. And the other one is homologous to this one. So when they are aligning themselves, it is a random orientation. Random, it is not specific that they align themselves in this side. So because of that aligning themselves, what is going to happen when we are getting the gametes? There is a possibility that the big one, which is red, this one, can go with the smaller one, which is red. Or the big one, which is red, can go with the smaller one, which is blue. Or it can be the blue, which is long, can go with the blue, which is short. Or it can be the blue, which is long, can go with the red one, which is short. So that is what we are referring to as random orientation. When they are orienting themselves, it is known that we shall only get the red and the red going together. Or we shall only get the blue and the blue going together. There is also a possibility of a mixture where the red will go with the blue and where the blue can go with the red. So that is what you call random orientation. That is during metaphase 1. There is an equal chance. There is an equal chance of the resulting gametes containing either of the pair. There is an equal chance of the resulting gametes containing either pair of the chromosome. The red can go with the red, the blue can go with the blue, and still the red can go with the blue, or the blue can go with the red. So there is that equal chance of the resulting gametes containing either of the pair. As humans, for humans, we have 23 pairs of chromosomes. Therefore, there are 223 gamete recombination. There are 223 gamete combination. If you get all our total number of chromosomes, which is 46, and that makes a pair of 23 pairs of chromosomes. So when we are forming a gamete, the combination is random. Each of the chromosomes can go with either. So when you look at the potential gamete combination, we can get a combination of up to 223 gamete combination. Very many combinations of gametes. So, orientation of pairs of homologous chromosome prior the separation is random. The orientation of the homologous chromosome prior separation is random. Here, when our chromosomes are going to align themselves on the spindle, their orientation is this one going to be on the left, this one on the right, this one on the left, this one on the right. It is random. So when you separate them, if they have arranged themselves in such a way, and you separate them at the center, this one goes the other side, the other one goes the other side. So you see that it is random separation. It's not that red will go on the same side and then blue goes on the same side. It can happen, blue going on the same side, red going on the same side. But there is also a good chance that red can go with blue, blue can go with red, or blue can go with blue, or red can go with blue. So that orientation of the homologous chromosome before the separation is random. And that one also is what brings about variation. Sources of variation still, we are now looking at gamete fusion. Gamete fusion. Now, during the gamete fusion, this one, the fusion of two haploid gametes, and they are from different parents. We are fusing two haploid gametes from different parents, results in the formation of a diploid zygote. And this diploid zygote contains genetic information from both parents. So if the information is coming from different parents, that means that each parent is bringing haploid gamete. And when you bring half the number of chromosomes and the other one brings half the number of chromosomes, then they will combine themselves together and form a diploid zygote. And now we are containing the genetic information from both parents. So that one is going to bring about variation when there is fusion of gametes. So if we have the egg or the ovum coming from the mother, and it is going to combine with the sperms coming from the father, and they are going to form a zygote, that is during fertilization. This one is going to bring a half, and this one is going to bring a half. They are haploid. The egg is haploid, the sperms are haploid. Then they will combine their chromosomes, and when they combine and they form a zygote, the zygote now is going to become diploid. Then the zygote is going to differentiate, and then it will form our embryo. Our embryo is now going to be distinct from other embryos, genetically distinct, different, because it has got genetic information from both parents. This promotes variation. This is going to promote variation as offsprings will be genetically different to the parents. Offsprings are going to be genetically different from the parent. Now, if we look at our embryo here, this offspring of ours is going to be very different from the mother and will also be different from the father. But is going to be amalgam. It will have, because this child of ours got half of the information, genetic information from the mother and half of the genetic information from the father. So he is going to be a recombinant. He is just genetically distinct from the parent. He is different from the parent because he or she has got information from both parents. So that's why we are saying this is going to promote variation as the offspring will be genetically different from the parents. So it brings about what we call variation. The fusion of the gametes from different parents promotes genetic variation. Because there has been fusion of gametes, and these gametes are coming from different parents, they have fused it together and fertilization has taken place, it is going to bring about variation, meaning that the offspring is going to be different from either of the parent. Will be different from the mother and will be different from the mother. So that fusion of gametes has brought about the variation of the offspring from the parents. We go to non-disjunction. Non-disjunction refers to the chromosomes failing to separate. When the chromosomes fail to separate, that is during anaphase 1 and anaphase 2 of meiosis, results in gametes with extra or missing chromosomes. And that one is what we call aneuploidy. So we saw that during anaphase 1, homologous chromosomes are going to separate and go to opposite poles. But whereby there is failure for those homologous chromosomes to separate, and all of them, they are pulled and they go to the same side. Now that one is going to cause what we are calling non-disjunction. And it will result in a gamete whereby one gamete will be having extra chromosome and the other gamete will be missing a chromosome. That is in anaphase 1. But when it comes to anaphase 2, when we are separating now the sister chromatid, sister chromatids in anaphase 2, they are the ones that are separating. And each sister chromatid goes on its own side pole, on its pole. But if the sister chromatids fail to separate and the whole chromosome, the sister chromatid, go on the same side, then that one is also going to result into what we are calling aneuploid, whereby one cell will have an extra chromosome and another one will be missing the chromosome. So that is what we call non-disjunction. It is whereby chromosomes fail to separate during anaphase 1 and anaphase 2 of meiosis, resulting in gametes with extra or missing chromosomes, what we call aneuploidy. The failure of the chromosomes to separate may involve 1. Homologous pairs in anaphase 1. all 4 daughter cells will be affected. If it takes place in anaphase 1, whereby homologous chromosomes fail to separate in anaphase 1, this will result in all the 4 daughter cells being affected. 2. If sister chromatids in anaphase 2 fail to separate, only 2 of the 4 cells will be affected. Only 2 of the cells will be affected. Because that one, it is taking place in anaphase 2, whereby we are separating the sister chromatids. If now in anaphase 2, one of the sister chromatids fails to separate, it will not affect all the 4 cells. It will only affect the 2 cells. So, non-disjunction can cause what we call Down syndrome and other chromosome abnormalities. It causes Down syndrome and other chromosome abnormalities. That is non-disjunction. If a gamete with an extra chromosome fuses with a normal gamete, the resulting zygote have 3 chromosome copies. And that one is going to cause what we are calling Down syndrome. And if a zygote has got 3 chromosome copies, then we refer to it as trisomy. And trisomy, Down syndrome always occur on chromosome number 21, where chromosome number 21 failed to separate and it got an extra chromosome. So here let's look at this non-disjunction overview. In normal meiosis, we have our homologous chromosomes here aligning themselves on the same spindle, and here they separate. This is in meiosis 1, homologous chromosomes are separating and moving to different pole. And in meiosis anaphase, this is anaphase 2, we are separating sister chromatids. Here the sister chromatids are separating and going to different pole. And we end up with haploid cells, 4 of them. All of them are haploid. These ones are normal. They are normal, with the haploid number of chromosome. Then we go to part 2, non-disjuncture. And this one is taking place in anaphase 1. Non-disjuncture is taking place in anaphase 1. Here, first of all, we have, before division, we have our homologous chromosome aligning themselves together. Now, when we come to anaphase 1, there is our non-disjunction. We can see that these chromosomes here, they are separating themselves. This one is going this way, and this one is going this way. But when you come to this second pair of homologous chromosomes, all of them, they refuse. All of them, they go on one side. So when we get now our cells, you will see that this cell here has got 3 chromosomes. And this side here, it has only 1 chromosome. So meaning that the one which was supposed to be here has moved to the other side. They are not equal. This one has 3, 1, 2, 3. This one has 1. So when we go to anaphase 2, Now here we have 3 sister chromatids separating themselves. And this side we have only 1 sister chromatid separating. So when we form our gametes, here you are going to see we are forming a trisomy with 3. This is also a trisomy with 3. And here, now it has only 1, yet it is supposed to be having 2. It is less. This one is more by 1, this one is more by 1, and this side, this one is less by 1, and this one is less by 1. Now, here, the non-disjunction happened in anaphase 1. And we say that when non-disjunction happens in anaphase 1, it affects all the gametes. It is going to affect all the gamete. This gamete has been affected by having an extra chromosome. This gamete has been affected by having an extra chromosome. This gamete has been affected by having less by 1. And this one has been affected by having less by 1. Because the non-disjunction here took place in anaphase 1. Now let us see another non-disjunction when the non-disjunction occurs in anaphase 2. In the beginning, before division, we have our homologous chromosomes moving together. We come to meiosis 1, anaphase 1. Here we are seeing that the homologous chromosomes are moving to different poles. This one is moving this way, this one is moving this way, and this one is moving this way, and this one is also moving this way, which is normal. Now when we come to meiosis 2, in meiosis 2 here, now we are going to separate the sister chromatid. When we are separating the sister chromatid, you will see that this one will allow the sister chromatid to move in different poles. This one will go this side, this one goes the other side, this one goes the other side, this one goes the other side. So that one is normal, there is no problem. But when we go to our second cell here, this one goes the other side and this one goes the other side, which is okay. Now when we come to this chromosome, these 2 sister chromatids fail to separate, and all of them, they go in the same direction, which is wrong. There is no one which has gone to the other side. So when we form our gamete, now here it means that now this gamete here has got 3 chromosomes, and this gamete here has got only 1 chromosome. Whereas if we come from those ones that came from here, we can see this one has got 2 and this one has got 2, which are normal. Normal, haploid, normal, haploid. But when we come to this side, this is abnormal. It has an extra 1. And this one is abnormal because it has a deficiency of 1. So here you find that only 2 cells have been affected. This one has been affected and this one has been affected. But when you go the other side, this one is not affected and this one is not affected. So these ones are normal. These ones are 2 normal. So the only ones that have been affected are only 2. So that's why we say that when non-disjunction occurs in anaphase 2, if non-disjunction occurs in anaphase 2, that is, it is going to only affect 2 of the 4 daughter cells. Non-disjunction incidences. Non-disjunction incidences. There is a positive correlation between maternal age and Down syndrome. So here we are saying that non-disjunction is likely to occur when the maternal age continues going high, we are likely to have Down syndrome risks. Now, if you look at the mother's age here, at the time they are getting pregnant, and you look at the risk of Down syndrome, when the mother becomes pregnant at an early age, the risks of having Down syndrome, they are also very low. But as the maternal age goes on increasing, so does the risks of receiving Down syndrome birth continues. So if you look at the maternal age going, for example, up to this point, if we try to extrapolate, you will see that also at this point, the chances of producing a child with Down syndrome, the risks also increases. So there is correlation between the maternal age and the Down syndrome risk. As the mother conceives when the age is going beyond 30s, the risks of having Down syndrome risk birth also continues. So maternal age is increasing, leading to higher incidences of Down syndrome. As the maternal age increases, the Down syndrome increases. And it has been seen as years have been going on from 91, 95, 2003, there was a high risk of Down syndrome babies went on increasing as the mothers went on producing babies at an age which is high. So you can see that the normal babies can normally occur below 30 here. If you look below 30, the risks of producing Down syndrome babies is low. All the babies are normally normal. But as the age go above the green line, and the mother's age is beyond 30 going upwards, you can see the risks of producing Down syndrome babies has been increasing as the years have been proceeding. From 91, the risks were lower, but they were existing beyond 30 years of age, and they have gone on increasing. If you go up to the statistics up to 2013, they increased highly up as the maternal age goes on increasing. Non-disjunction karyotyping. Now, karyotyping is the process by which chromosomes are organized into a karyogram. It can be used to identify non-disjunction events leading to aneuploidy. So we can use the karyotyping to organize the chromosomes into what we call a karyogram. And this karyogram is going to help us to identify the non-disjunction events which leads to aneuploidy. Aneuploidy, we said it is whereby the chromosomes fail to separate, and we have some gametes having extra chromosomes and others missing chromosomes. So when we arrange our chromosome and organize them into a karyogram, we can see where non-disjunction events happened by using a karyogram. Cells can be harvested from the fetus, and induced to undergo division when they are in a condensed DNA. So we want to see the non-disjunction. We can get cells from a fetus and we induce them to undergo cell division, and then we can see where a neuroploid has occurred. The chromosomes are then stained with a photographed to produce a karyogram. They are going to be stained, the chromosomes will be stained so that they are easily photographed so that we can produce our karyogram. Fetal cells can be harvested via different techniques. We can get these cells from the fetus in different techniques. There is a cryonic villi sampling. Here cells are extracted from the placenta tissue. We extract the cells from the placenta tissue. Then we have amniocentesis. Here the cells are going to be extracted from the amniotic fluid. Then we have chorionic villi sampling. Here the cells are extracted earlier, that is approximately at 11 weeks. But this one has a higher risk of miscarriage, which is approximately 1%. So here they are extracting the cells from the villi. They can be extracted from the villi, chorionic villi. But the problem here is that there is a higher risk of miscarriage if the cells are obtained from the chorionic villi. Then we have amniocentesis. In amniocentesis, the cells are collected at approximately 16 weeks. And these ones, the cells are collected from the amniotic fluid. It has a lower risk of miscarriage, around 0.5%. So the cells will be collected from the amniotic fluid, and then they can be induced to undergo division so that we can see whether a neuroploid has taken place. Examples of aneuploids. We have examples of aneuploid. We have Patau syndrome. Patau syndrome, a trisomy occurs on chromosome 13. If we have 3 chromosomes here, that would be Patau syndrome. If we have 3 chromosomes on chromosome number 13. Edwards syndrome, trisomy occurs on chromosome number 18. That is Edwards syndrome. Edwards syndrome, if it occurs at chromosome 18 and we have 3 chromosomes here, that would be Edwards syndrome. Then we have Down syndrome. Down syndrome, it occurs on 21. If you look here on 21, we have 3 chromosomes. You have 3 chromosomes, 1, 2, and 3. So we have a trisomy. Now here our karyogram is telling us that a neuroploid has occurred on chromosome number 21. A neuroploid has occurred on chromosome number 21, and when it occurs on chromosome number 21, trisomy 21, then that one is Down syndrome. So this individual, when you look at the sex chromosome here, you will see that this is XX, so this one is a female, but she's having trisomy at chromosome 21. If she's having trisomy at chromosome 21, now the total number of chromosomes this individual is having is 47 chromosomes. But the normal number of chromosomes is supposed to be 46 chromosomes. So this means that this individual has got an extra chromosome, one extra chromosome. And that is what we are referring to as the trisomy, which is taking place on chromosome number 21. They are supposed to be homologous chromosome, 2 pairs, but now they are 3 chromosomes together. So if we find that we are still looking at examples of aneuploid, Klinefelter syndrome. This is whereby now an individual has got an X chromosome and also a Y chromosome. So if you come to the sex chromosome here, and there are 3 of them, there is X and then another Y. That is called Klinefelter syndrome. It is also a neuroploid. If the individual has only one X, that is now called Turner's syndrome. If you come here, instead of having X and X or X and Y, and the individual has only X, one X, monosomy X, that one is what we call Turner's syndrome. Aneuploids tend to occur on shorter chromosomes. Aneuploid tends to occur on shorter chromosomes. So you see, it is always taking in a range of 17, 18, 19, 20, 21, and maybe the sex chromosome. It always occurs at shorter chromosomes. Hello, my friend. Thank you very much for participating in meiosis. And today we have looked at the sexual life cycle. We have looked at the gametes. We have talked about the stages of meiosis. We have looked at crossing over. We have looked at random assortment. We have talked about non-disjunction. And we talked about the karyotyping. Thank you. Please don't forget to subscribe.